Okay, folks, I think we'll get started with the symposium. Well, welcome back, everyone. I'm Lee Kump. I'm the Dean of the College of Earth and Mineral Sciences at Penn State. And I'm pleased to be the moderator for the symposium this morning. Marcia mentioned that there are two areas that seem to engage the public. One is in planetary science and space, and the other is the oceans. And we're focusing today on ocean health. We're midway through what UNESCO has designated as the Ocean Decade, one in which there's to be a focus on the science we need for the ocean we want. And in fact, we do want and need a healthy ocean. The ocean provides a wealth of services to human society and to natural ecosystems, ranging from carbon storage, and we'll be hearing more about some consequences of ocean carbon uptake later on in the discussion today, to coastal protection, food, livelihoods, tourism, natural products, and a sense of place. And indeed, the ocean's health is in decline. Pollution from human activity on land is running into the ocean, impacting coastal zones, triggering harmful algal blooms, and creating dead zones. Fossil fuel emissions are increasing the atmospheric CO2 level, driving climate change. A significant fraction of that CO2 has been absorbed by the ocean, and that's driving acidification. The diversity and structure of coral reefs is in decline for these and a multitude of other reasons. Warming is further strengthening the ocean, stratifying the ocean, and that, together with reduced oxygen solubility in a warmer ocean and increased microbial rates of decomposition, is depleting the ocean's interior of oxygen. Simultaneously, we are considering expanding our exploitation of the ocean's resources, namely through deep-sea mining. So UNESCO charts a course for us for the Ocean Decade and beyond, from identifying stressors, their distribution, sources, and variability, to understanding the biological response, exploring adaptation and mitigation strategies, and translating this new knowledge into policy actions that will lead to a healthy ocean able to sustainably provide those services that are so important to society. To explore these multiple stressors more completely and discuss what can be done to address them, we've established a group of researchers with particular expertise in each of these 5 areas, but as you'll see, with notable interdisciplinarity among them, expertise ranging across the range of stressors and impacts, because there are multiple stressors impacting each of our valuable ecosystems across the ocean. So we'll hear from each of these folks individually and then engage in a dialogue about what the future bodes under various scenarios, whether we should anticipate tipping points of no return, and what needs to be done now to minimize the impact of these stressors on the ocean. So what we're going to do is we're going to hear from each of our 5 panelists first, and I'll ask you to save your questions for the open Q&A at the end. We've gone through the 5 speakers. We'll take a little break with a really engaging short video from the Bigelow Laboratories, and then we will engage in a panel discussion followed by open Q&A. So to start things off, I would first like to introduce Jacqueline Padilla-Gamino. She's Associate Professor in the School of Aquatic and Fishery Sciences at the University of Washington. Jacqueline got her Bachelor's of Science degree in Oceanography from the Universidad Autónoma de Baja California in Mexico, her Master's of Science in Biology from Cal State Northridge, and then her PhD in Oceanography from the University of Hawaii at Manoa. Dr. Padilla-Gamino studies the ecophysiology and reproductive biology of marine organisms in a changing environment. By combining field and laboratory techniques, she examines the importance of transgenerational effects in acclimatization and local adaptation, and investigates the synergistic effects of multiple stressors on coastal ecosystems. She is an International Coral Reef Society Fellow, a recipient of an NSF Career Award, and a Sloan Research Fellowship. So let's welcome Dr. Padilla-Gamino.
I'm very excited to be here and share some of the work that I do and talk to you a little bit about some stories that are happening in the Pacific Northwest. Thank you so much for the invitation. So the planet is getting warm, oceans are getting warmer, and this is just a graph showing how the last 3 years are the warmest in the recorded history. And we also just got a 10 out of 10, right? The 10 warmest years in the historical record has been the last decade. So things are getting hot. And in addition to that, the same carbon dioxide that is driving atmospheric warming is also reshaping the chemistry of the oceans. And the oceans have acidified around 30% since the beginning of the Industrial Revolution. So you can see on the right graph values of CO2. So on the x-axis you have time, on the y-axis you can see CO2 in the atmosphere on the red, with the red line, which is like our longest time series for CO2 at Mauna Loa atmospheric CO2 station. And on the green color, you see the CO2 in the ocean. That's also our longest record of CO2 at Station Aloha through the HOT program. And you can see how as CO2 is increasing in concentration in the atmosphere and in the ocean, there is a decrease in pH shown by the blue dots. So this basically means that the waters are getting also more corrosive. And this is due to several chemical reactions that occur in the ocean. As CO2 comes in, it combines with water. Then you have carbonic acid, and this carbonic acid is not very stable. It dissociates between carbonate and hydrogen ions. And so that's why the waters become more corrosive, because now you have more hydrogen ions that lowers the pH. So that's the first problem. The second problem is that the hydrogen ions will interact with this carbonate molecule. And this carbonate molecule is really important for building shells, but it will react and form more bicarbonate. So that's the second problem, that we no longer have the carbonate that the organisms need to create their shells. So not only are waters becoming more corrosive, there's also less carbonate, which is an important building block for any organism in the ocean that has a skeleton, exoskeleton. So what can organisms do against change, right? Some of them can move, so they can migrate. Others cannot move, and that's problematic, right? Corals, mussels, oysters, they cannot move, so they have to deal with it. So they can acclimatize, which basically means adjusting their physiological conditions for the change. They can adapt. This happens over multiple generations, and is the population that adapts by constantly changing the DNA pool over multiple generations, or they can go extinct. So those are the choices. You move, you adjust, you adapt over multiple generations, or you die. So that's important to keep in mind. And so today I'm going to talk a little bit about the future of seafood, which is really at the front line of ocean change, because a lot of them require this calcium carbonate that I talk about to build their shells. And so this is really important because some of them now will require more energy to produce their shells, and some of them might not even be able to produce them in specific sites. And that's a problem for a multi-billion-dollar global industry that depends on the production of these organisms. That's important for future food security around the world, and also is important for cultural and traditional values that rely on shellfish organisms. So I'm going to tell you very quickly, just mention really 3 stories in the Pacific Northwest associated with warming and ocean acidification and shellfish. So in 2021, we had this massive die-off on the intertidal zone in Washington and Vancouver, and this was due to a heat wave event. It happened to be around 120 degrees Fahrenheit or 49 degrees Celsius, but it happened to coincide with low tide and midday. So basically the shellfish, all the organisms that depending on mussel reefs or mussel beds died. So basically they cooked. And there's nothing they could do, there was no way to run. And we project that more of these events are gonna be happening because we see that heat waves are getting longer in duration, more frequent, and have higher strength. Another example was with the oyster shellfish growers. In the 2000s, there was massive devastation of this industry because the baby oysters or young oysters within the first 48 hours could not really survive. And part of that was because they could not build their shells. So that's when scientists and shellfish growers start developing partnerships to monitor the water chemistry. And they realized that the reason why they were not surviving is because the water was too corrosive, right? The regular water that has been used for decades to grow the oysters was now too corrosive for the youngsters to produce their shells. And so now there are strategies. Now they can add some chemicals to the water so it's not corrosive, so that they can continue to grow. But this is another massive influence of the change in ocean chemistry on the production of an important product. And then in terms of mussels, some mussels rely on attachment. Otherwise, water can just take them away. And they do that through these called vessel threads. It's like strings that have some glue. They're quite magnificent. I love them. But so this glue, these molecules that the vessel threads use, are sensitive to ocean acidification. And so that's a problem, for example, for mussels that are attached to rock and use these vessel threads to deal with the waves. And it's also a problem for this shellfish grower that depends on attachment to these lines to have a good harvest for the next year. Because now, under ocean acidification, the mussels cannot attach well, and so they lose their product. They fall from the lines, and and that's a problem. And besides talking about organisms with economical importance, there are foundational species. There are ecosystems engineers that are having a really bad time with ocean warming. So on the left side, you see a beautiful kelp forest, and on the right side, a magnificent coral reef. Dr. Jackson will focus more on the coral reef, so I'm going to tell you a little bit about the kelp forest. So kelp forests have a complex life cycle. Sometimes they are macroscopic, sometimes they are micro. And unfortunately, around 1/3 of kelp beds around the world have declined. And specifically in the US, those declines have been more dramatic, around 96% in Northern California, 65% in areas like Puget Sound in Washington. And many organisms depend on them. So if we don't have them, we don't just lose the kelp or the reefs, but many organisms that depend on them. But in terms of the life stages, what we have found is that they are sensitive. The big stages, the macroscopic stage, right here, is sensitive mostly to temperature. But what we have found is that the other stages, these little guys here, which are microscopic, actually are very sensitive to ocean acidification. So you have a complex life cycle, and many organisms do have a complex life cycle in the ocean, and different stages will be vulnerable to different stressors. This is just an example of a short experiment we did with the microscopic stages. In different colors, the lines represent different temperatures, and in terms of pH levels, a solid line is a higher pH level, a dotted line is a more corrosive water, right, lower pH. So on the left panel, you see growth. And it's very interesting how growth happens the best in acidified waters at high temperatures, which is what you don't really want. But that's when you have the highest growth. But if you see the right panel, the exact same organisms are crashing in terms of reproductive success, right? So this is what we need to study. It's not enough to study one stage, but multiple stages to really identify the critical bottlenecks that are limiting the survival and persistence of organisms in the sea. And in addition to that, understand the stressors. Where are they doing their best? Where are they doing the worst? And how does that change through the seasons? Because this is physiological capacity to adjust is dynamic, it's not static, and changes depending on where you are and the season you are. And once we know that, then we can really know the effects of multiple stressors, right? How is this curve of performance can actually shrink or not if there are more and more stressors. Because ocean acidification and ocean warming, I believe, are two of the most important factors of ocean change, but we have many more, like plastic pollution, nutrients. Now we have more fires in the coastal zones. And so there's a lot of work to do, a lot of gaps, and a lot of new things that we need to understand. Thank you.
Next, we're going to talk about ocean deoxygenation, and we're fortunate to have Brad Seibel, a professor from my alma mater, PhD alma mater, the College of Marine Science at University of South Florida. Brad received his Bachelor's of Arts degree in Biological Sciences from the University of California, Santa Barbara, and his PhD in Ecology, Evolution, and Marine Biology from that same institution. Dr. Seibel employs a unique suite of field and laboratory techniques and approaches to assess the ecological consequences of climate change, including ocean acidification, deoxygenation, warming, and the role of animal energetics in ecosystem dynamics. He's a fellow of the American Association for the Advancement of Science, and he's been the chief scientist of over 20 oceanographic research cruises. So let's welcome Brad Seibel to the stage.
Well, thank you, Lee. There's no other environmental variable as important to marine ecosystems that has changed so dramatically in such a short period as oxygen. That statement was written 30 years ago by Diaz and Rosenberg in their classic review of marine hypoxia, and it's clearly just as important today as it was then. These images show examples of the effects of deoxygenation on marine habitats around the globe, including on the left phytoplankton blooms in the Arabian Sea that have completely altered the ecosystem there. Fish kills are becoming a regular occurrence around the globe, and even coral reefs are susceptible to massive die-offs during hypoxic events. The ocean has lost about 2% of its total inventory of oxygen over the last 50 years. And this map shows some hot spots in the dark red where they've lost actually about 2% of the oxygen per decade over the last 50 years. Models suggest that this deoxygenation is going to accelerate and continue well into the future. Ocean deoxygenation includes both coastal hypoxia and oxygen minimum zones. These two habitats are different in many respects, but they also share some commonalities that are worth considering together. Oxygen minimum zones are indicated in blue on this map. So these are large regions of low oxygen at intermediate depths from about 200 to 700 or 800 meters in some cases depth in the eastern upwelling regions of ocean basins. Oxygen minimum zones have expanded over the last 50 years by an estimated 4.5 million kilometers squared, roughly the size of the European Union. Coastal hypoxic zones indicated in red are clustered around human populations and point sources of pollution, and these have more than doubled in the last 30 years. There are 500 sites indicated here. When Diaz and Rosenberg wrote their paper 30 years ago, there were fewer than 300. So ocean deoxygenation is caused ultimately by a mismatch between oxygen supply and demand. In coastal environments, the primary cause is eutrophication. Agricultural, industrial, and human waste literally fertilizes the ocean. The phytoplankton produced ultimately sink and are decomposed by microbes that consume oxygen, leading to these dead zones in bottom waters in coastal environments. Ocean warming is the primary culprit for the open ocean. Ocean warming reduces oxygen solubility in seawater. It increases stratification that prevents mixing with the atmosphere, and it increases the oxygen consumption rate of resident organisms. And the ultimate result are dead zones and expansive oxygen minimum zones. One of the largest dead zones in the world is located in the Gulf of Mexico here. It reached a record size, roughly the size of New Jersey, a few years ago. And it's driven by more than 1 million tons of nitrogen pollution that flow from the agricultural belt of the United States through the Mississippi River each year. Hypoxia can completely alter marine ecosystems very rapidly. So healthy coral reefs and kelp forests and seagrass beds are lost and replaced by microbial mats. Mobile organisms, fishes, crustaceans, and squids can sometimes escape and relocate. Less mobile organisms, like the brittle stars shown in the bottom right, leave their hiding spots and come out in search of better access to oxygen. Low oxygen affects the habitat quantity and quality for species that are of interest to fisheries. We call it habitat compression, where low oxygen is forcing species into narrower, localized areas, and this can have the effect of making them more vulnerable to fishing and overfishing as fishermen target these areas. In other cases, it forces them further offshore and increases the effort required to catch. Habitat compression is also a major concern in the open ocean. As the oxygen minimum zones expand, the breadth of them increases such that the minimum oxygen levels are shallower and fishes are forced into shallower and shallower water. Diving depths are limited in some of these large predators like billfish, tunas, and sharks, such that either they're physiologically limited or the prey that they seek is forced into shallower water so that they're not required to dive as deep. In any case, the prey are more vulnerable, and these large predators are more vulnerable to fishermen that target oxygen minimum zones. This same sort of habitat compression affects vertical migrators. In the open ocean, zooplankton, fishes, and squids migrate on a daily basis. They spend nighttime at depth of about 300 meters, nighttime up near the surface where they feed, and daytime at depth of about 300 to 500 meters where they're avoiding visual predators. So they're basically staying in the dark. The the expansion of the oxygen minimum zone forces them shallower on their deeper end of their migration. And in the most extreme oxygen minimum zones, they're also limited by light. There's a limit to how shallow they can go before they're more vulnerable to predators. And so they have adapted in a way that allows them to suppress metabolism and stay at depth in a sort of state of diapause. That's what you're looking at here. These squids, which are normally extremely active, voracious predators, are effectively asleep at depth during the daytime in the eastern tropical Pacific. Small differences in oxygen over a relatively small horizontal region. So, sorry about that. So over a few kilometers distance, that variation in oxygen there leads to important differences in the abundance of some of these migrators. So we've known about the importance of oxygen in physiology and ecology for a very long time. It's been very thoroughly studied, and yet there is no widespread agreement on how to define hypoxia or how to define hypoxia tolerance. So what are the safe oxygen levels for marine ecosystems? In humans, a relatively small change in oxygen leads to important changes in cognition and motor function. Learning and spatial memory are impaired, and when you get to about 50% oxygen, which is the equivalent of the top of Mount Kilimanjaro, we're talking about loss of consciousness and ultimately death. In contrast, in the ocean, anything above 50% is considered normal. That's normoxia. We define hypoxia primarily by metrics that are unsustainable physiological endpoints, like the critical oxygen partial pressure, indicates where aerobic metabolism is no longer able to maintain even basic maintenance. Loss of equilibrium indicates a complete failure of neuromotor function. And we use LC50, lethal concentration curves, where 50% of the organisms in a trial are killed. And this has led to widespread adoption of extremely low oxygen limits that are considered okay. So recent work suggests that marine animals are not all that different from humans, and that we've sort of overestimated their hypoxia tolerance. We can put animals in flumes like this one and exercise them, basically a treadmill for fishes, and we can change the oxygen level as they swim. As you see here, at greater than 100% air saturation, metabolism is unaffected by oxygen. And as soon as you get below 100% saturation, you get a linear decline in aerobic capacity and aerobic scope. That's the amount of oxygen available to support growth and reproduction and predator-prey interactions. If you are an animal that's specifically adapted to low oxygen, the slope of that line will be increased, and the critical point, the break point in that line, will be lower. We've measured these indices in a variety of species now. And what you can see is that for shallow living marine animals, for the most part, they fall on that line, the dashed line there. It doesn't matter what temperature, what body size, or what species you are. No matter what your metabolic rate is, you require 100% saturation to achieve your capacity. Anything less than that, like humans, you begin to decline very rapidly. Species that are adapted to low oxygen, such as estuarine species shown in blue, they require about 50% saturation to achieve their entire capacity. So they don't care about oxygen until it gets to 50%, and then it declines rapidly. Species in the oxygen minimum zone are more variable because they're found at different depths in different regions, but anything less than about 20% begins to affect those organisms. So the take-home message with that is that the critical oxygen pressure basically for all species is the current oxygen level. Any decline in oxygen pressure will impact marine animals. And so the real question becomes, what is the duration and the intensity and the frequency of hypoxic events as much as the oxygen concentration we focus on? Thank you.
Okay, thanks very much, Brad. Next, we're going to talk about harmful algal blooms, and we're fortunate to have Don Anderson with us. He's emeritus research scholar at the Woods Hole Oceanographic Institution. Don found MIT early on in his career, has his bachelor's in mechanical engineering from MIT, a master's in and PhD in civil and environmental engineering from MIT as well. Dr. Anderson's group studies toxic algae responsible for disrupting human and ecosystem health, including red tide organisms. His research ranges from molecular and physiological studies of growth, sexuality, and toxin production to the large-scale oceanography and ecology of the blooms of these microorganisms, including numerical modeling, forecasting, and a range of monitoring and management strategies, many reliant on novel instrumentation and biosensors. He has won numerous awards, including the NOAA Environmental Hero Award. Let's welcome Don to the stage.
Well, thank you, Lee, and good morning. So, as Lee said, we're talking about harmful algal blooms, or HABs, as we refer to them. Some of you may know this as red tides. These phenomena are linked to algae in the freshwater and in the ocean. There's some beautiful pictures of algae here. There's tens of thousands of species of algae in the ocean, and the same for seaweeds. They're critical to the air we breathe and the food we eat, but among those are a few hundred that cause harm in different ways. And I've listed just some of those impacts here. At the top, you see these human poisoning syndromes that are associated with shellfish or fish that have accumulated these toxins from algae, but through the food web. And they all have these acronyms with different types of poisoning effects. Amnesic shellfish poisoning causes permanent loss of short-term memory. I won't go into other details here, but they're all serious illnesses, and a number of them can be lethal. In addition, we have animal mortalities. You've probably seen pictures just like the one that Brad showed of dead fish. Many of these millions of dead fish on beaches are caused by toxic algae, and the same for marine mammals, whales, dolphins, manatees being killed by these. But also at the bottom, you see there's impacts from non-toxic species in blooms. And one of those I've highlighted is that associated with seaweeds. These are HABs. And I'm going to this morning try to give you just two examples of these impacts where I see and others see that we've reached a tipping point, places where there used to be no problem or little problem, and now we have massive impacts and probably little likelihood of reverting to the former state. So, as I said, let's start with the seaweeds. Some of you have heard about these sargassum inundations, which are incredible millions of tons of seaweed washing up on beaches in the southeastern U.S. and throughout the Caribbean. For me, when I think of sargassum, or when I first thought about it, I remembered grade school and hearing about the Sargasso Sea. And this is an 1891 image showing, it was made for basically sea captains to see where they are likely, those are probability lines, where they're likely to run into the masses of sargasso or sargassum in the North Atlantic. And I use this to point out that red oval that I inserted at the bottom, which is where we now see the origin of these incredible phenomena that are going on these days. It wasn't there before. So this was recognized, though, in this paper in Science, and they named it as the Great Atlantic Sargassum Belt, the largest algal bloom on Earth. You can see that it stretches from the west coast of Africa all the way across the Atlantic and through the Caribbean and into the Gulf. And this is easily visualized in satellite imagery, and so this same group has gone back, and on the left you see a small video going through year after year the distribution of sargassum in that North Atlantic. And I've made these snapshots so you can more easily see some of the progression. And the first thing that you can see here in 2003, that this looks like that Sargasso Sea image that I showed you from 1890. Notice in 2010, though, this has extended and even reached the Iberian Peninsula and the west coast of basically Northwest Africa. Both areas of intense upwelling, a lot of nutrient-rich waters there, but also, as you'll see, currents that bring water down the coast of Africa. And then you go to 2012, now we're seeing the first filaments of the seaweed down here coming from that region, and look how much it's intensified by 2018. Now we see the same thing up here in the top where you see, again, remember satellite imagery is perfect for looking at this. So you see nothing up in here until you see 2010, 2011, then you see some impact, and then this has continued to the present day in huge abundance. So really what happened? The general consensus is that a warming climate, a warming ocean has changed ocean currents, has changed weather patterns, and as a result, this seaweed has been able to propagate over to this coast, this side of the Atlantic, catch the nutrient upwelling here, take the ride on these currents down into the tropics, where then it has this not only abundant sunlight and warm waters, but incredible nutrient inputs. You don't see it here, but coming from the Congo, coming from the Amazon and some other rivers, adding a huge nutrient source, in part because of the deforestation and the farming that's going on. These are major forces that drive nutrient inputs into the coastal zone. So what we end up with then are these incredible inundations that are happening throughout the region. You can see that a recent estimate is just the state of Florida alone spends $3.6 billion annually to try to clean up this problem. And again, think of this as a tipping point, something that wasn't there before, that's happened, and it's hard to imagine how it will revert to the former. All right, let's move on to this paralytic shellfish poisoning, or PSP syndrome. And this is one that is the most widespread of all of the poisoning syndromes we have in the HAB field. You can see it all over the world. In North America and many other places, it's caused by an organism called Alexandrium catenella. That's what we'll be looking at now, but I've circled up there the waters north of North America and the Arctic, and notice there's no red dots up there. We have no real information about what's going on. So when I started to look at this, notice here in the Gulf of Alaska and southeastern Alaska, we have a lot of knowledge about this problem up there. It's been around a long time, but essentially no history and no knowledge of this region of the Bering Sea and the Chukchi Sea or even the Beaufort Sea over here. And why do we want to look there? Well, this figure pretty much says it all. Over here you see the temperature increase through time from 1900 to the 2020s for this little green polygon up here, which we call the Chukchi Sea, right north of Bering Strait. And you can see that it's warming up 4 times faster than the global average, global ocean average. Why? It's pretty simple, that the sunlight, when it shines on ice and snow, most of the energy is reflected. Once that melts, most of the energy goes into the water. So that differential gives you this amplification of the warming that everyone else is experiencing. So then you have to ask, what is it doing to this organism, Alexandrium catenella, that we know is up there? This is a diagram showing its life history, and these are resting stages or cysts, like seeds of terrestrial plants. And that organism is in the sediment most of the time. But then when the temperatures are favorable, they germinate and give us the bloom of the vegetative cells, produce the toxin, and that's really where the problem is. And then they make new cysts that fall back to the sediments. And one of the first things you do when you study up here, this is my lab's data, we start mapping out the distribution of those seeds. This shows you the potential for bloom in that region. And honestly, this is one of those moments in science where you see something that just blows you away. This is astounding to me. And the reason is, you see here, this is the Gulf of Maine. Nova Scotia and Boston, Woods Hole's down here. And for years we've been studying this annually recurrent outbreaks. That's drawn on the same scale, both density and area as here. And you can see this area that we thought was too cold and there was no problem has 20 times the distribution of this organism cis. And then it's no surprise whatsoever that then when we start having cruises and looking in the water, we see these massive blooms that are massive, not only in stretching 1,000 kilometers or more, but having incredibly high toxicity as well. So this is up here in the water column. So what we've come up with is a conceptual model of what's happening up there. And basically, let me go back here. What we see is that as the Bering Sea, it's already warm, but it's getting warmer, that water moves up, and the blooms that establish themselves here moves up through this jet, this northward flowing water through the Bering Strait into the Chukchi. So if you look at it on the side, you see these blooms coming in, dropping these cysts out to bottom sediments. But historically, it's been really cold down there, too cold for these cysts to germinate. So you get a lot of inputs, no losses. What do? You generate that enormous cyst bed. I called it a sleeping giant in the PNAS paper we wrote about this, waiting to emerge. And now what we're seeing is the temperatures there are warm enough for this entire cycle, for germination, blooms, new cysts, then being augmented by these transported blooms. So it's really an incredible situation, and you have to ask, what does this mean to all the communities up there? They have no history of this. We keep asking, have you got this in your history? Do you eat things differently during certain times of the year? And the answer has been no. So what's happening? Well, the answer has to be that it's an extraordinarily dangerous and worrisome situation. And the reason is that the communities up there are subsistence harvesters. They don't go to the grocery stores. They collect everything from the sea or from land. And so where we understand very well for the rest of the world how this toxin moves through the shellfish and other crustaceans, we know very little now about this. We get asked, can I eat my seal? What tissues are safe in a walrus? Can I eat a seabird? What about the seabird eggs? So we're struggling to come up with all of these answers, all in a region that didn't have this before. So another tipping point here to highlight. So just a few final thoughts. I've told you that HABs are extraordinarily diverse. I've only showed you 2 of these indications of the kind of impacts that can be out there. We've already heard the oceans are changing in many ways, whether it be pollution or climate or warming, and HABs are changing as well, like I'm showing you here. Many are getting worse and spreading to new areas. And perhaps more in the context of this panel, some HABs have reached these tipping points. They're either approaching them or they have actually exceeded them, and where we don't see any likelihood of reversion to those former conditions. And finally, there's obviously a significant need for the new tools and technologies for management, monitoring, and potentially even control of some of these. I haven't had time to talk about it, but would be happy to if someone's interested. So thank you very much.
Great. Well, next we're going to talk about coral reefs, and we have Jeremy Jackson here who is going to lead us through that discussion. Dr. Jackson is emeritus faculty member at the Scripps Institution of Oceanography and the Smithsonian Tropical Research Institute, and he's a research associate at the American Museums of Natural History. There's a bit of a homecoming, I guess, for him. He has his bachelor's and master's degrees in zoology from George Washington University and his PhD from Yale in geology. Dr. Jackson studies threats and solutions to human impacts on the environment and the historical ecology and evolution of tropical seas. He is a member of the United States National Academy of Sciences and the American Academy of Arts and Sciences, and has won numerous international prizes and awards. Let's welcome Dr. Jackson to the stage.
When people ask me what I do and I say I work on coral reefs, the inevitable response is, oh, that must be really sad or really depressing. And the fact is that it's been pretty grim for about 25 years. as we've watched the exquisite systems I studied as a kid degrading dramatically. But what I hope to convey to you in these few minutes today is that it's not all bad news, and that there is, in fact, evidence of evolution and adaptation happening in coral reefs, which we should be paying more attention to instead of the latest grim reports. Work, please. The big button, yeah, okay, thanks. So coral reefs are actually a very small part of the ocean, really about 1%. I think of it as coral reefs as the scum on the rim of the bathtub. But they have enormous economic and social importance. They feed millions of people in the tropics, and shortages of food or breakdown of coral reefs cause mass starvation in many countries. More than a quarter of all marine species live in this very, very narrow zone of the ocean. They also provide billions of dollars through tourism and fisheries, protect coasts, et cetera, et cetera, et cetera. The key thing to understand about coral reefs, and we've really only known this for about 20 or 25 years, is a coral isn't just a coral. So, a coral animal consists of a colony, typically, of a lot of polyps. Think of them as being like miniature sea anemones. And that animal is inhabited by a very diverse biota that really determines the function of corals. The little brown dots that you can see in the polyp are dinoflagellates, symbiotic dinoflagellates, termed zooxanthellae. The zooxanthellae fix carbon in the form of sugar, feed the junk food to the corals, and the coral's survival is utterly dependent on the sugar produced by the zooxanthellae. The coral returns the bargain by providing nutrients and a certain amount of amino acids. And what you all have heard of as coral bleaching is the breakdown in the partnership between the coral animal and the dinoflagellates that live inside their tissues. But the coral also provides some of its precious fixed carbon in the form of mucus to the surface of the polyps. And we used to think the mucus was there mostly to be sloughed off and keep the coral polyp clean. What we now know is that the mucus is a virtual ecosystem of microbes. Something on the order, a piece of coral the size of my fingernail supports somewhere between 800 and 1,000 species of bacteria, viruses, and fungi. And those organisms, among other things, fix nitrogen, essential for protein synthesis, and they produce antibiotics, which are vitally important for the health of the coral. The health of corals depends on the balance of power of the microbiota that lives on the surface of the corals. And coral disease represents a situation in which, say, something like Vibrio explodes in abundance because of an imbalance due to any one of a number of things. Now, what everybody hears about is coral bleaching, and we refer to bleaching. Bleaching is the turning white of corals, which occurs because the zooxanthellae are lost from the coral when temperatures get too high. Coral holobionts live very close to their thermal maximum. So temperatures of 2 or 3 or 4 degrees above the normal maximum for a period of months wreak absolute havoc, and the corals kick out the endosymbionts, which no longer are providing the sugar, and they die if they can't get new symbionts within about a month. Heat waves, as you all know, are increasing in frequency and severity, and mortality is commonly greater than 50%. For terrestrially bound creatures like everybody in this room, the way to think about it is imagine how you would feel if you woke up one morning in August and learned that 80% of all the trees on the east coast of the United States had dropped their leaves. And then you read a month later, 50% of all those trees that dropped their leaves died. Well, that's what happened to the corals on the Great Barrier Reef, which is a length of coastline roughly equivalent to the entire east coast of the United States, when 50% of the corals in the bleached areas died. This is a really, really big deal. But it's not just climate change, and this is the great intellectual battle which is roiling in the coral reef community between the people who say it's temperature, 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 and then some of us others who say, well, yeah, but it's also all the other stuff besides the increase in temperature. So, for example, this slide shows the severity of bleaching in relation to the amount of nitrate that's in the water at different sites around Moorea after a moderate heating event. And what you can see is, so the y-axis is severity of bleaching, the x-axis is the amount of total nitrogen in the water, and the severity of bleaching, whether the heating event is low, moderate, or extremely high, the proportion of corals that bleach and the severity of bleaching is a linear function of the amount of pollution in the water. And this is just one of the many, many aspects of coral health that are affected by the local environment that in relation to human activity other than the rise in temperature. This network of interactions gives you a little bit of a feeling for what's going on. Bleaching and global warming, high temperatures is in the bottom right of the figure, and corals dying is in the center. But you can see there are all these different things which affect the health of corals. Overfishing of herbivores results in abundance of seaweeds. The seaweeds rot and die. They make detritus. That carbon feeds bacteria. Those bacteria, many of which are pathogens, kill corals. Pollution directly causes coral deaths. Pollution increases the impact of bleaching. Pollution increases the abundance of pathogens, which kill corals over and over and over. This entire network, due to what we have been doing for a very long time, is what is really going on in terms of coral health. Think of it this way: if you could wave your magic wand and you could make climate change disappear tomorrow, coral reefs would still be in extreme danger of extinction because of all the other stuff on this slide, which is going on every day. And let me tell you, we are doing virtually nothing about all of those other things, particularly in this country. Okay, now the saving grace, or the potential saving grace in terms of all of this, is that there's enormous variability, natural variability, which exists among populations of all species of corals. A simple way of thinking about this is that warmer places have more heat-tolerant corals than cooler places. And this can happen on very small spatial scales, like the back of this room and the front of this room can have totally different thermal histories, and the corals that live in those two different places are able to play the game of life in very different ways, because they might be acclimated to higher temperature. Acclimation usually involves switching the different kinds of symbiotic dinoflagellates among forms that are more or less tolerant of higher temperatures, or adaptation in terms of heritable genetic differences among coral hosts. So I thought I'd show you one simple experimental example that was done by Morikawa and Steve Colombi. They went to American Samoa and they looked at two different places that were only 1 kilometer apart. And in one of those places, the red color is about, well, it has a lot more hot days, and it's a little bit warmer on average than this cooler place, which is only 1 kilometer away. So they collected little nubbins of corals from the hotter place and from the cooler place, and they took them to an intermediate location, and they planted them in a common garden, and let them grow, and they thrived in this place. And then a little bit later, there was a heating event. And when you assess what happened after that heating event, you look at the percent that bleached, and you can imagine many of those died. The corals that came from that slightly warmer place exhibited very low percentage of bleaching. That's for 4 different subspecies of corals, which you can see in this figure. Whereas the corals that came from the somewhat cooler place experienced much more extreme coral bleaching, as high as almost, well, 70% or so. Okay, so that's a cute little experiment. How does it scale up? And we're starting to see examples of this. So what I'm showing you here is the history of coral reefs in the tropical Eastern Pacific. Think of the shore of the Pacific coast of Panama or Costa Rica or Ecuador. And back in the 1980s, the coral reefs of the Eastern Pacific are sort of monotonous looking. They're not the picture postcard places that you all go to, if you do, but they have had very high coral cover, close to 50%. But then there was a big El Nino, a big heating event that occurred in 1980, and that abundance crashed to something like 1 or 2%. And for a decade, for more than a decade, there was almost no recovery of the corals. Then the stars indicate different bleaching events. And then around 2000, there was another big heating event, and the corals sort of whatever, and then they exploded. And by the time there was a third event, in 2016 and 2017, the corals went, oh, no big deal. And coral cover is now higher than it was way back then in 1980, before the first bleaching event. And then in 23, 24, there was another extreme event, and the data aren't all in, but I thought I'd show you the picture, which is on the right, because as you can see, and this extends for kilometers and kilometers, everything you see in that photograph is living, healthy coral. So what's going on in the Eastern Pacific? Clearly, it's not like the Great Barrier Reef. We think what's going on is 2 things. Well, one thing we know is going on is that the corals, after somewhere around the late 1980s, dumped one kind of zooxanthellae and got new ones. But now we're also starting to see adaptation in the corals. Even in the Caribbean, which has a grimmer history, coral reefs in the Caribbean, the ones I grew up on, are in really bad shape. So to cheer myself up, I went to Curacao in May of last year because I'd heard that the coral reefs in Curacao were still really good. And these photographs were taken in May last year. And you can see on the left abundant star corals and elkhorn coral. The picture on the right is a small forest of elkhorn. In the middle, lots of massive corals. Coral cover in those photographs is on the order of 50%. And lots of those corals are really large. So what's really interesting about that is, they're at least 2 or 3 decades long. old, and in that period of time, they have suffered lots of extreme heating events, and they've also experienced invasions of a horrid disease with the awful name of stony coral tissue loss disease, which has devastated, for example, corals in Florida. And that disease and bleaching had severe effects early on. But what we're looking at now are the ones that somehow survived that. And so these reefs are eventually seem to be turning into forms which are really not that much affected by high temperature. Overall rates of mortality have decreased with each event. It is not unimportant that this location with these beautiful reefs is at the eastern end of Curacao. Nobody lives there. It's a protected area. There's limited fishing. The water that comes in, bathes those reefs, is beautifully clear and full of food. When corals bleach, they can actually survive if they can eat, and corals eat zooplankton. So abundant food is also a big part of this question. So to sum all this up, I think this is what I'd like to leave you with. First of all, there are still billions, if not trillions, of corals in the ocean. They are not all gone, although their abundance is, of course, declining fast. And we all know that climate change is worsening. But biological systems are not set. Dobzhansky famously said that nothing in biology makes sense except in the light of evolution. And I would argue that coral ecologists have sort of forgotten that. They think about what the corals can cope with now. They think of them as static systems, but biological systems are not static. Natural selection during an extreme heating event could not be more intense. And in fact, we know from the Morikawa and Palombi experiments, and from now there are dozens, this is a growth industry, people doing this kind of research, acclimation and adaptation are occurring, and new and more resilient holobionts are evolving. And we're seeing that in places like the tropical Eastern Pacific and in Uspunt, in Curacao, and in a few other places. And research on coral reefs, I would argue, really needs to sort of make a change in focus and pay much more attention to this evolutionary potential instead of ever more refining the obituary of coral reefs. A paper was just published this year about the consequences of the big 2016-2017 bleaching event with like 50 or 60 authors. They have refined coral death, to the second decimal point. But we've known for a long time that a lot of corals died. And I would argue that we really need to put our energies into understanding this remarkable capacity for adjusting that nature has instead of just giving up. Thanks very much.
Thanks, Jeremy. Okay, our final presentation today is on deep sea mining, and we're fortunate to have Beth Orcutt here with us. She's the Vice President for Research and a Senior Research Scientist at Bigelow Laboratory for Ocean Sciences, and she's a research professor at Colby College. She did her bachelor's work in interdisciplinary studies at the University of Georgia, and her PhD in marine sciences also at the University of Georgia. Orcutt is a globally recognized expert in the environmental impacts of deep sea mining. She's authored some of the most highly cited papers on understanding the ecosystem services provided in the deep sea by microscopic organisms. Her expertise in deep sea mining impacts stems from over a decade of leadership experience directing diverse and international teams of scientists and students in the exploration of the deep sea. Using seafloor exploration assets and deep sea observatories, her research has focused on discovering how the deep biosphere, microscopic life, thrives in a strange environment and how their living impacts global processes. She's a recipient of numerous honors and awards, most recently the AGU Ocean Sciences Voyager Award. Let's welcome Beth to the stage, please.
It's a pleasure to be here with such a distinguished audience, and thank you, Marcia, so much for your service as president of the Academy. Our presentations today have really talked about the part of the ocean that we see when we go to the coast, right? The water and what's happening near us. And I think we often forget that the majority of the hard surface of our Earth is seafloor, and it's very diverse. There's mountains, there's trenches, there's a lot of different geography. And in those places, there's also a lot of rocks. And as Lee was mentioning, I've built my career studying a lot of the life that lives on those rocks and the interesting chemistry that happens there. In case you don't think about the deep sea very often, I want to remind you of some of these habitats that are down there. So this is a kind of cartoon representation of how I think of some of those rocky environments in our ocean. Starting on the left-hand side, where you have mid-ocean ridges or maybe a hot spot where you have magmatic processes heating up water that's interacting with the rocky environment and releasing lots of metals and sulfides into the water, and those precipitate as metal sulfides around these hydrothermal systems. When that hot water movement ceases, those metal sulfide deposits are often referred to as inactive sulfides. In those environments, especially in the hot water environments, you have really interesting chemistry that organisms can take advantage of to fix organic carbon. So like here on the surface Earth, we're very familiar with photosynthesis, right, and using energy from the sun. In those environments, you have microbes using energy from chemistry to be able to fix carbon and feed into that ecosystem. You also have really interesting symbioses happening between those organisms and animal life. So some of the animals in those environments like farm these microbes in their guts, and that's their food source. As you move away from those ridge systems and hydrothermal systems, you can still have these underwater mountains, seamounts. In those environments, you also can have interesting chemistry occurring where you have microbes that are able to use that chemistry to fix organic carbon, again, supporting part of the food web. And then in other parts of the seafloor and the abyssal plain environments, where you have very little input of organic material from the surface ocean, there are conditions that occur that allow precipitation of metals slowly over hundreds of thousands to millions of years to form what are called ferromanganese nodules. They're basically like my fist size, potato size lumps of black material. And these nodules also support really interesting animal ecosystems in those environments. Increasingly, there's more human interest in these environments. So there are some who are interested in collecting some of the rocks that form in these environments for the metals that they contain. So from the polymetallic sulfides that form at mid-Atlantic ridges, you might have things like copper and other metals that are precipitating. Likewise, in the ferromanganese nodules, there's also metals that are forming there. And there's interest in maybe trying to exploit that. There's also interest in using the rocky seafloor as a place to store carbon and injecting carbon-rich fluids into basaltic rocks and forming carbonate, and that's kind of illustrated off to the right. All of this is happening in the context of an environment that we actually don't know very much about because it's really hard to study. So a paper came out last year led by some colleagues of mine where they looked at all the video data that has been collected from human-occupied vehicles and remotely operated vehicles and co-located in geography, like where did those things happen, to show that humans have only had access to sea less than 1% of our seafloor. Here's one of those robots that's used to do this kind of work, showing some really amazing giant sponges in parts of our deep sea. Likewise, we haven't fully described all the different species that are down there, and some estimates are that we know less than 10% of the species. And here's imagery of an octopus garden that was discovered, some cruises that I was participating in, supported by the Schmidt Ocean Institute, and we'll be showing a video about that at the end of this. So there's really a lot that we don't know about the habitats that are down in this deep sea. And one of the most important things that we don't know is how are these species and communities connected? This is a picture of a baby octopus that we saw being born on the seafloor, and we were able to follow like 150 meters up into the water as it was starting its life and leaving the place that it was born. It was a very visual example to me of how hard it is to understand those different life stages, right? We heard about those, how important it is to understand the complicated life stages of organisms in the ocean. We don't know a lot about how sponges, coral, other organisms that live in the deep sea, what their life cycles are like. How endemic are they? If you cause a disturbance 100 kilometers away, will that matter? And so this is something that needs more research. To put this in context, for us here in the United States, I think we often forget that when you add up the territory of the United States, our country is more than 50% ocean. And that's because of the exclusive economic zone that is around many of the territories in the Pacific that are claimed by the United States. And most of that is in the deep sea. So that means that we actually don't know a lot about the life that lives within our country's territory. And this is a map that was put together by NOAA a few years ago. It doesn't actually show the full extent of our current exclusive economic zone. Overlaying in that context is also where might these rocks be that are expected to have these high metal contents. And there's basically three mineral types that are discussed. These ferromanganese nodules that are shown in the blue color that I was talking about. Those similar kind of crusts that form on seamounts that are really, really old. You see that in kind of the salmon color. And places where those might co-locate is kind of shown in yellow. And you can see that that overlaps with a lot of those territories in the Pacific. And there's increasing interest in looking in those places and potentially exploiting those resources. As Lee mentioned at the beginning, I've been part of trying to take what we know about the deep sea and put it in the context of how would you describe this as a service that humanity gets and that the broader community of life on Earth gets. To remind ourselves that even though these ecosystems are far away from our everyday experience, they do provide value for evaluating, is that service maybe more important or less important than the economic value we might get from collecting those deep sea minerals. So some of those services include supporting climate regulation. They are providing habitat and trophic support. They're involved in the cycling of a lot of other chemicals in the environment. Because of how little we know about the life down there, that also means that there's a lot of genetic diversity down in the deep sea. And that genetic diversity might be the source of things like new antibiotic discoveries and new enzymes that have industrial potential, as well as potential sociocultural service. Lee mentioned, and I've mentioned too, that there's an emerging concept that humans might try to mine some of these minerals from the deep sea. And this is a schematic representation of one approach where you would use a collector vehicle that's connected to a riser pipe that moves along the seafloor, trying to pick up some of those materials, send them up to a ship. The ship would dewater that material and send some particle-rich, dirty water back into the ocean because they don't want to transport that back to land. So the types of impacts that scientists are trying to understand and the companies that are interested in these activities are trying to understand is whether that particle-rich water or the plume that is distributed on the seafloor from that disturbance, whether it has toxicity, and that could be acute toxicity or chronic toxicity. It's very hard to study this or to translate what we know about ecotoxicity in the surface world to the deep sea. It's a very different energy environment. So that's a question. There's a lot of filter feeding organisms down in the deep sea, the corals, the sponges, et cetera. Will they be smothered by these particles? And will they be able to clear that and keep growing, or will that be a mortal impact? There may be noise pollution, so sending up all those rocks up a riser pipe might create quite a bit of noise, and noise can travel, sound can travel very far in the ocean. We know that from how animals communicate with sound, that sound can travel quite far distances depending on its frequency. There may also be light pollution associated with these activities depending on the technology that's used. And then as I mentioned, if you're removing rocks, you may be removing habitat, you may be creating conditions where you have a loss of overall diversity if you're losing species. And so that's a big question. The need to understand this question has accelerated quite a bit in this last year. So this is a screenshot of Executive Order 14285 that came out last year, directing the federal agencies here in the United States to really accelerate understanding the deep sea and the minerals in the deep sea to inform the potential exploitation of those minerals. Just yesterday, I was visiting with some of our federal colleagues to discuss this. And really, part of that science is to understand what are the biggest knowledge gaps that we have right now to help understand these ecosystems and whether or not this activity would be harmful. So first of all, we actually don't know if the minerals on the seafloor have the metals in economic quantities that would make sense to even go get them. So those maps I showed you were perspective maps of where these metals could be, but we actually don't have a ton of data to validate that, and that's something that colleagues, for instance, at the USGS are trying to understand. Then at the biological level, we've seen so little, we've sampled so little, that our understanding of the biodiversity in the deep sea and how widespread that biodiversity is something we don't know. And I am very biased towards seeing the deep sea because that's where the rocks are, but we also have to remember that there's water above those rocks and that there's also biodiversity in the water above that could be affected. Jeremy gave us some great examples of ecosystems that have resilience. Does the deep sea have resilience? Could it survive those kinds of impacts? Could it survive cumulative impacts of not only the deep sea mining, but also increasing changes from climate change and things like that? We don't really know that very well. And importantly, because this is such a remote ecosystem, we really need to accelerate our ability to develop cost-effective strategies, to develop baselines, and then do monitoring. We can't go see whether or not deep sea mining is working. It really relies on trust and sharing of data from those ecosystems for us to know that. So we really need to push on understanding those baselines and developing these tools. The National Academies of Science, Engineering, and Medicine was requested to stand up a committee by Noah Bowman-Bessie to recommend a research strategy to try to close some of these knowledge gaps. I am serving on that committee right now. I encourage you all to go look at the statement of task for what this group needs to work on. And I think that's my last slide. Yes, so lots going on. We're trying to fill these research gaps very quickly. I am so grateful and I feel very privileged that I've had the chance to go to the deep sea and experience it. And I wanted to leave you with a little bit of that joy and excitement of seeing the deep sea. So I think coming up next, there's going to be a short video here that was put together from some footage that was collected from a cruise in 2023 supported by the Schmidt Ocean Institute, where we discovered some of these octopus nurseries offshore Costa Rica, to remind you of the inspiration and awe and beauty of the deep sea as we go into our panel next. So thank you very much.
Well, thanks for sharing that with us, Beth. Really amazing video. We're just going to have a bit of a discussion up here and then save as much time as we can for your questions. So please bear with us a bit. Just a couple areas that I think we could pursue a bit more before we turn it over to the group for questions. And that is in thinking about the ocean decade and the call to science. One group of questions are around the science gaps and the need for additional research. The second part will be what can we do? But thinking about that first part now, we've seen that there are a variety of stressors. They're intertwined. Two questions related to those. Are there other stressors? Have we cataloged all of the stressors that exist that are impacting ocean ecosystems? And are there ways to disentangle those in terms of their biological and ecosystem impacts? And then we talked a little bit about tipping points. Are there ways to anticipate tipping points before they or as they're developing? And are there points of no return? So with that as a general question around need for additional science, maybe we'll just turn it over to the group here. Maybe I'll start with you, Jacqueline. Your thoughts on this needed science gaps?
Yes, you hear me?
I think so.
Yeah, so definitely a lot to do still. When I heard your question, I immediately thought about phthalates. So phthalates are chemicals that come from, they're basically plasticizers, chemicals that come from the plastic. And there's a lot of plastic in the ocean releasing these chemicals, and we don't know exactly how to even measure them. Right? Or if we do, it's really hard, very time intensive. And so just sometimes figuring out the levels of pollution or the stressor can be a challenge. But without even thinking about synergies or antagonistic interactions. Like sometimes it's just about identifying them and measure them and just being aware of them. And so, for example, phthalates are endocrine disruptors that can affect development, reproductive processes. And as plastic pollution continues to increase, it has increased dramatically these last 80 years. So I think it's identification of the stressors, understanding patterns, and then understanding how dynamic is the capacity of the organisms to adjust and whether there is adaptation potential. So that I see it. And for some stressors, we know more, but for others, it's just even figuring out if there are, for example, hotspots of these chemicals.
Well, Brad, you were talking about deoxygenation, but you couldn't help but talk about other factors that are there. So what are your thoughts about these multiple stressors, disentangling them? And
I think that they are hopelessly entangled, and temperature in particular is one that I think is entwined with hypoxia tolerance and misunderstood in a lot of ways. I think, clearly as the oceans warm, the oxygen in the environment decreases. But all of the experiments that we do are short-term acute exposures in the lab. And I think this follows on what Jeremy was talking about, that there's bound to be some resilience and plasticity in the responses. So if you increase the temperature, you get an acute response in metabolism, and depending on how you measure it, an apparent loss in hypoxia tolerance, but measured over seasons, that disappears often. And I think we need a lot more long-term experiments and a lot more seasonal and multi-year and evolutionary sorts of experiments.
Don, I know you have additional thoughts about multiple stressors, and you've given us a great example of a tipping point realized. So your further thoughts on this?
Sure. The one that comes to mind in light of what Jacqueline said is ocean acidification. And people often say, oh, what's going to happen to HABs in a more acidic ocean? Well, they're just algae, and when you have acidic conditions, you have more CO2 in the ocean, therefore all the algae are going to have, that's part of photosynthesis. They need that. But I liken it to if you fertilize your lawn, you get more grass, but you also get more dandelions and clover and weeds. And so we expect, in the simplest level, yes, we will see the HABs more against the background, also elevating of the beneficial algae. But we also know that some of the HAB species that produce toxins produce more toxin under acidic conditions. And simultaneously, we know we can limit some of our species with phosphorus, for example, and make them 10 times more toxic than they would be if we limited them by nitrogen. So if you combine an acidic environment with one that is also a low nutrient, you can have much, much more toxins. So there's a huge synergy there that we are just only beginning to understand in terms of the multiple stressors.
Well, Jeremy, it all comes together around coral reefs. It seems everyone had a coral reef picture in their presentations, and you yourself pointed out, for example, and I follow the coral listserv, and I've seen the arguments, about warming and bleaching and nitrogen. I just wonder if there's anything else that you'd like to say, about this from from any perspective.
Well, I think we could go on cataloging new things, but I think the real need for looking into this more carefully is to look at what happens when you remove those stressors. So, I mean, since it is in theory within our capacity to reduce nitrogen input or whatever, I mean, the city of St. Petersburg did it because there was this massive eutrophication in Tampa Bay, and it became really disgusting. And for reasons that had nothing to do with biology and everything to do with the aesthetic of living on the shorefront, they really cleaned up sewage in that area. And it had this extraordinary effect, and seagrasses came back and all the rest of it. We need a lot more of that to show that it is within our capacity to regulate the stress environment, I think, in all marine ecosystems.
And then, Beth, you're sort of in an interesting place in the deep sea because there's so little known. Maybe, cataloging the stressors, we don't even know the organisms down there. Yet we're thinking about significant human activity there. So, what are your thoughts about, from a deep sea perspective?
Yeah, I'll maybe share this one perspective that I think maybe brings it also closer to our human experience, and that is that some of the areas that are being discussed for this potential activity of deep sea mining may increasingly overlap with areas where highly migratory fish species are that are part of highly profitable fisheries, like tuna fishing, example. And that those fisheries may be moving due to warming and climate change and other stressors. And so you may have increasingly overlapping areas where these activities may be co-located. And there's concern from some of the fishery management organizations that if could there be metal magnification up the food chain into those predator species. We know that already happens with, for instance, like methyl mercury and certain types of sport fish, et cetera. And so there's concerns about could there be metal remobilization that feeds into the food web and then that gets amplified up in these species that we eat. And so you have to understand all of those pressures and how they might be changing in addition to what is happening on the seafloor for thinking about these new industries.
Fantastic. So I think that the next point is around action. What steps should be taken, could be taken to address the threats to ocean ecosystems that we've been discussing today. And so, Brad, maybe I'll start with you. Any thoughts about next steps, beyond the science?
I think there's a lot of activities that could be done in order to reduce especially coastal deoxygenation, different management of agricultural practices and cover crops and those sorts of things that are outside of my area, but I'm told there are a number of sorts of management practices that can be instigated that have been shown to be effective in a lot of areas. In the open ocean, it's a much more daunting case where we have to stop producing CO2. How do you do that? But certainly starting with the coastal, and as Jeremy mentioned again, the case of Tampa Bay is a success story, and by just changing some sewage treatment practices, Tampa Bay was turned around. So I think it's the best place to start.
Don?
Well, the history of problems in my field in terms of trying to control halves has been rather poor. It's been so much easier to get funding for fundamental science for all these years, and yet I've come down to DC and testified multiple times, and I'm constantly asked by staffers and members of Congress, what are you going to do about it? And we get that from the public all the time. But when you write proposals and say, I'm going to study the genetics and the physiology or toxin chemistry, that's much easier to fund than saying, I'm going to throw something into the ocean to try to kill this organism and not hurt other things. But what's happened in my field is that we've actually created programs specifically on prevention, mitigation, and control with their own funding lines and separate calls. And that has been hugely successful at bringing lots of people into that area. Years ago, I wrote a little commentary in Nature because I went to a conference where there was one talk out of 500 at a conference I went to about bloom control. Now, if that was an agricultural conference, it would have been something completely different. So what's happened finally is that the field is getting there, but it is now requiring a special attention from the funding agencies to target, and from the states. The state of Florida has an $18 million 6-year program on control of the Florida red tide. So that's generating a lot of nice technologies and good approaches. So I could give you specific examples, but it's more to have the national and state level support for a level of practical science that isn't coming at the expense of fundamental science. That's the key thing. We can't raid the ecological and physiological and oceanographic programs to fund these practical studies. We need them both.
Jeremy, that description of Curacao was encouraging, and I think about intervention versus protection, or reduction of harm. So and I know in the coral listserv again, there's different the transplanting of corals as an intervention. Just curious about your thoughts about. And you've talked a little bit about the steps needed to take to address coral reefs and some promise from resilience and adaptation evolution.
Well, one of the really interesting developments that's going on has to do with trying to determine the results of genetically mixing corals from very distant places, and it's turned into this. Well, for example, cryopreservation of sperm. Because you can't very well take a coral from Florida, put it in a bucket, and fly it to Curacao. But if you can freeze the sperm of the coral from one place or another, then you can do the experiments. And the limited amount of work that's been done in this area is really interesting. There is, I'd be shot for saying this in another room, but there is a sort of sense of a hybrid vigor that comes because these corals have been isolated from each other for a very long period of time, right, by current patterns and all the rest of it. So I think this is actually a really exciting frontier to see whether or not we have the capacity to accelerate processes of adaptation. And the initial results are really interesting. There are lots of other things, but that I think is an example of a very creative new kind of science But yeah.
Beth in the deep sea. You know, I guess part of this is, you know, regulation of deep sea mining, exploration, exploitation. But you know, what are these next steps, and who's in charge?
Yeah, that part I'm not going to touch. What is needed? Something I failed to mention is the scale of this potential activity. So if we're thinking about those nodule areas in the abyssal plains, some economic studies have suggested that any given lease for mining would need to mine an area of about 120 square miles a year to be economically viable. And since we've used Tampa several times as an example, that's the size of Tampa. You would have to mine that area every year for every contract, every leaseholder. So that's the scale of the potential removal of habitat and the potential impacts into the water column. Science is not caught up to that scale of understanding biodiversity and projecting what would you need to protect to ensure that if you're causing impact, you haven't lost diversity, you haven't lost function. We have no idea if these ecosystems can recover from that damage. The experimental mining that's been done decades ago, you go back to those habitats, they look like they were freshly impacted. To get at that scale, we need to have way more vessels and commitment to do deep sea science. We're losing vessels out of our academic fleet that are aging out. And they're not being replaced. So this is going to require like massive levels of investment in infrastructure to be able to understand the science and the baselines to even inform the industry. So that's where my heart is focused right now.
And Jacqueline, of course, you know, thinking about acidification itself, it's sort of like Brad's challenge, you know, for the open ocean deoxygenation. It's tied to the buildup of CO2. But there have been some geoengineering thoughts about adding alkalinity and what are these next, what are the next steps and what are the long-term prospects for the impacts of the acidity of the ocean?
I'm not as familiar with those technologies, but what I think is outstanding about the Pacific Northwest is this partnership of scientists and shellfish growers. I think we need more of that to solve any problem associated with ocean health. Start with people. And so in the Pacific Northwest, I think that's where I've seen the most collaboration, which is very, very positive, I think. I study a lot thermal stress and acidification, which are associated with the CO2 problem, right? And it's very difficult to engage people and say this is happening because they cannot see it. But shellfish growers can definitely see their oysters dying. And if you will have been in that major heat event that I talk about, you will have smelled everything dying on the beaches. And so I think identifying those examples or engaging in different ways can help us to have more power to solve many problems. And one of the reasons why I shift a little bit focus on plastic pollution in my lab is because it's more tangible. Everybody knows what plastic is. We are all covered by it. It's in our houses, our kitchens, our bedrooms, in baby supply products, and nobody wants, no matter what your party is, right? Like nobody wants trash. And I think that and that's another CO2 problem, right? The plastic. So yeah, I just want to say that. Like I think working with people, engaging, finding ways to connect is a solution. Yeah.
All right, folks, we have 15 to 20 minutes for your questions. We have microphones on both sides of the auditorium, so please queue up and we'll just go side to side to those microphones to answer your questions. So, and we have names here so you can address your question to particular people or to the panel as a whole. So we'll start over here on the on your right. Yes.
Bill Clark, Harvard University. So the focus here on the multiple stresses across the ocean system was just delightful to hear, really exciting. I wanted to retreat from that. Actions we take tend to be on one dimension rather than the multiple stress dimension. Were you to be asked, or were the Academy to be asked, how much the damage scenarios that you've worked out here would be mitigated were society to be implementing the Paris Agreement goals for reducing fossil fuel emissions enough to keep climate change within 1.5 to 2 degrees? How much reduction would that cause in the damage suite that you've been talking about?
Just let anyone who would like to respond.
I can
I
say
think for coral reefs, it would make a big difference because the magnitude of the heat stress events would be, wouldn't keep going up. But I would argue that we'd still be in trouble if we didn't address all the other things.
there's a modeling study that concluded that going back to pre-industrial CO2 levels, within a few centuries, shallow water oxygenation would return, but deep water would take on the scale of centuries to millennia to recover. So, that depressing note.
I would have to try to figure out just how long it would take that change in the atmosphere to propagate into the ocean, and especially to the bottom of the ocean. But if I use that Alaska example, we have a function we use that tells us how long it takes those cysts to germinate at different temperatures, and it follows an 85-degree day function. And what that means is that if it's 10 degrees in the bottom, it'll take 8.5 days to germinate. But if that shifts by 2 degrees, let's say it gets colder, you can see you go from 6. It's a change in the amount of time it takes to germinate that can be from weeks to even a month, depending on where you are. So you could actually start delaying germination of those cysts to the point where in those high latitudes they can't bloom. There's not enough light and so forth. So a couple degrees propagated to the deep water could make a huge difference.
I would answer that question like, I have no idea because we know so little. Some of the places that we study in the deep sea, like we've maybe seen once. We have no idea if there's seasonality, how dynamic those ecosystems are. So my analogy is like, imagine going to a forest in the wintertime, and that's what you think the ecosystem looks like. So how would you know what the climate impacts would be? Because you don't even know how that ecosystem functions on a yearly scale. So we have a lot farther to go, I think, before we understand that.
Okay, we'll go over here.
Sure. Jim Elser, I'm from the University of Montana, and I'm a limnologist, so I'm here to speak on behalf of inland waters. So inland waters face many of the same stresses that were described by the panel, including nutrient oversupply. I just want to make a brief policy comment. Farm practices were mentioned. That's our main problem, is agricultural practices right now. The biggest policy issue there is agriculture is exempt from the Clean Water Act. And if we want to do one thing to address these issues, we would bring agriculture under the Clean Water Act, and that would get you kicked out of every congressional office in Washington, D.C. very, very quickly. So that's the power problem we have, is the proper action that's needed is politically impossible at the moment, but maybe things can change. I do have a question for Don, though, because you made a good list of all the impacts of eutrophication, harmful algal blooms, but one thing that I think is concerning to me and becoming more prominent is the aerosolization of toxins from harmful algal blooms. And I just wondered if you could describe that to for the audience and tell us what you know about the status of that work these days. Thank you.
Sure, thank you for that question. There's two ways to answer it. In the ocean, there are aerosolized toxin problems, the most prominent one being along the coast of Florida, where if you get an onshore breeze, that particular toxin gets airborne in sea spray, and people can't breathe as they walk along the beach. The lifeguards have to wear masks, and you have to move inland and so forth. And there's another organism we have in the ocean that, in the Mediterranean in particular, causes all these flu-like symptoms of people along the beaches. So it's a very common, although in most cases transient problem. I'm actually more worried about your freshwater world and the aerosols there, because there has been a study up near in the lakes of New Hampshire where they're finding clusters of neurodegenerative diseases among the populations that live close to bodies of water like lakes and so forth. And we know the cyanobacteria, the freshwater blooms, the green scum you often see, are prominent in many of those lakes. And a lot of concern now is that there's airborne transport of those cells, and therefore their toxins, that people are just inhaling without any knowledge of the impacts. And so I think a huge area of future research, especially in the freshwater world, and there are people looking at this, is the inhalation of a lot of these freshwater toxins, and especially the risk to people that live near some of these water bodies.
Yeah, Dan Kamen, Johns Hopkins University in Section 64. And I have a question for Beth, but maybe just a preview piece, is that there's this biannual Our Ocean Conference taking place. The next meeting will be in Mombasa. It will be in June, and it's the first time it's been in Africa. And so we are running a workshop the week after, June 20 to 24, trying to particularly get young researchers who are not necessarily been working on oceans their whole time, on marine energy, on bioplastics, on many of the issues here. And so if anyone's interested and has kind of a student at the postdoc, grad student level that's interested, we'd be keen to. We can support a few more people coming to the meeting. So the question for Beth is that you're always looking for ways to kind of highlight the issues like what's not seen can't be observed in the deep ocean, and seabed minerals is a critical case of that. The statement that we know more now about the dark side of the moon than the deep ocean comes up a lot. One of the areas where I do feel like there's an amazing discovery that hasn't gotten as much notoriety outside of the deep ocean community is we've now discovered inorganic processes in those nodules, so-called dark oxygen. And if oxygen is being produced inorganically on the seafloor, and we are also permitting in front of the International Seabed Authority these trial contracts to start harvesting these materials, right when we are discovering amazing new things, we do not know what the ocean impact is. It seems like we have one of those kind of aha, nova, 60 minutes type of moments to highlight we really need a moratorium, and yet we have this group in Jamaica, the ISA, going ahead and essentially permitting the metals companies and others. And so I wonder if you can talk both a little about the science, but also, is this a lever we can use to slow down what looks like a incredibly damaging and unnecessary from the perspective of the materials side of the equation? So.
Yeah, thanks for your question. I actually got into the field of deep sea science because I was inspired by the idea that there might be life on other planets. And I thought that the deep sea was one of the best places to study how does life survive in weird places. And your mention of the theory about dark oxygen production, I think is really intriguing because it scratches an itch of, is there other chemistry and other energy forms that support life that we have overlooked because we haven't been asking the right questions, and that would inform our search for life in other places. I'm really inspired by the team that's trying to study that, and I know they have a lot of cruises coming up to really try to verify the theory that that's happening. Whether or not those types of discoveries rise to the level of stop considering use of the deep sea for human activities, I think is a really interesting societal question. I think that goes along hand in hand with the question about, do we actually understand the economic viability of collecting these materials? What are the drivers? If there's geopolitics involved, as you mentioned. So I think, from my point of view and where I sit, it's part of reminding us about how much we still don't know about how the planet that we live on and that supports every human that we love, how it exists, and what is important to study and learn. while we're also having those really important societal conversations about what we use from our oceans. Thank you.
Okay, over on this side.
Rama Govindarajan from the International Center for Theoretical Sciences in Bangalore. So my question is about ocean fertilization as a strategy to increase carbon sequestration. How seriously is it being taken? How likely is it to become a reality? And is it a good or a bad idea for the future of the planet?
That's beyond my pay. Oh boy.
I'll take a little stab there. In terms of how it's being considered, it's an idea that's been around a long time. It started with John Martin many years ago when he said, give me a rusting tanker or something like that, and I'll give you an ice age, something like that. But the idea of fertilizing the ocean with iron scientifically, it's attractive, because for every atom of iron you might add to areas of the ocean that have a lot of nutrients and low iron, you get like 10,000 or 100,000, a huge uptake of carbon. And so recently there are now, there's a whole community that's going at this very, very carefully. I've got some colleagues that are funded to try to model this, because one of the issues is you can add iron and the phytoplankton grow, but what happens to that carbon? If it gets consumed and recycled and before it reaches the bottom, you haven't sequestered anything. So you now need to know how do you can't just do those simple experiments like they did years ago. Now you've got to be putting in the sediment traps and modeling this. But there is a major initiative. I know at my institution they're looking at it very, very hard. And I think it's justified, because the payback is quite large for that. But they are being very careful. They're being much, I think, appropriately careful. So I can't comment on whether it will work. I can just say that the process is, they're moving ahead scientifically, I think, in a deliberate and intelligent way.
Brad, any comment about ocean interior oxygen consequences from iron fertilization?
I think you'd have to be very careful where you chose to try these fertilization experiments. Certainly anywhere near the coast, that's phytoplankton's going to end up on the bottom and create dissolved oxygen issues. But I think if the research is being approached carefully, as Don mentioned, then it's worth pursuing. I did work a long time ago on carbon sequestration, where we were proposing to take liquid carbon dioxide and put it on the bottom of the deep ocean, and we were doing some small experiments to prove that that was a bad idea. I think is the way I approached it anyway. And I actually thought that both fertilization and carbon sequestration had sort of just been abandoned some like 15, 20 years ago, but they've reemerged because we haven't done anything else. And so it's on the table again.
I'll maybe just add a short perspective on this, in that and thinking about the question we had earlier about the Paris agreements, right? So I think our interest in this is driven from the data that we need to get carbon out of our atmosphere to avoid these climate change impacts that we don't want. And just stopping fossil fuel use, which is the biggest thing we need to do, isn't enough. And so that's really driving this interest. And I would, to connect this to the topic of deep sea mining, I have heard in discussions that some of the mining companies have asked, like, could we get credit if we put this metal in the ocean and it caused a fertilization bloom? And like, maybe that's part of our economic model, is the carbon markets. So I bring that up in this context because I think we don't want to be siloed in our thinking about it as just ocean iron fertilization, but how it connects to all of these questions in the ocean.
Yes, over here.
Alan Hastings, University of California, Davis. Apologize, this might be a bit off the question. Is that it's almost the elephant in the room. I've heard no comments about the potential for large changes in ocean circulation and that what effect that may have on everything that we've heard about today. I mean, AMOC is one area, but other possibilities certainly exist, and there were suggestions that these are not zero, far from zero probability events in the lifetime of people alive today.
shutdown of the AMOC. Any comments about this? Certainly, data that are being used to drive models that would suggest a significant decline in the vigor of the overturning circulation in the Atlantic.
I'm trying.
I will say I was reminded recently of an interesting story in the Arabian Sea. I showed a picture of the phytoplankton bloom there. That seems to be caused, as I understand it, from a loss of ice in the Himalayas, where that's driven a temperature differential, and so we're getting wind blowing in a different direction, creating different circulation and upwelling of nutrient-rich waters, and that's leading to deoxygenation and that phytoplankton bloom that's completely altered the ecosystem. So certainly there are changes in ocean circulation, and I'm not brave enough to comment on the AMOC situation, but it's a possibility.
And I'll take the same approach. I don't have an AMOC. I understand I don't have a linkage. But in the northeastern U.S., another area that's been touted to be warming more rapidly than many others is the Gulf of Maine. And one of the reasons that's been hypothesized is that the behavior of the Gulf Stream is changing, and it's always sort of in a battle against the Labrador Current that comes down from the north, and that Labrador Current has typically brought this cold water into the Gulf of Maine. A lot of lobster and algae and so forth thrive on that. But as the Gulf Stream is changing its behavior and more of these warm eddies are spinning off and coming into the Gulf of Maine, it's making dramatic changes to all of the fisheries. And so a few years ago, it was a big situation. Everything was warming. Now we just had a speaker at a recent symposium who talking about how it's cooling down again. So it's the erratic behavior of some of our major ocean currents that are affecting specifically the region, and in my case specifically some of the HABs we study. It's not one directional right now where I'm talking about in the Gulf of Maine, but the changes are significant. It's big temperature changes in a very short period of time.
And another example of that is the failure of the upwelling in the Gulf of Panama that occurred a year ago. First time in 100 years. The entire structure of that ecosystem is driven by the seasonal upwelling as the convergence moves north and south. And the wind stopped blowing across the isthmus, and the upwelling stopped, and the fisheries went to practically zero. And the corals, for the first time in the Gulf of Panama, bleached because it was the first time they had ever experienced hot water, which all the other parts of the Eastern Pacific had. So that was huge, and we have no idea whether that's going to happen again.
I guess, interconnectedness for sure, and slowing's one thing, but we've emphasized tipping points, and an abrupt shutdown might be something. Well, we've seen Hollywood showed us what happens when the AMOC shuts down abruptly, and it's not a pretty sight. So something that we need to be adding to the list for sure. Over here, maybe for our, try to get through these questions very quickly here.
Peter Palese from Mount Sinai in New York. We heard that the Great Barrier Reef in Australia is in bad shape. Is there any evidence that it repairs itself? So in other words,
Heals itself.
The Great Barrier Reef in Australia, we heard that there are big problems with the corals there. Is there any evidence that it repairs itself?
Well, they have to stop dumping sewage into the Gulf of Aqaba to make any difference. I've been there. I worked briefly with a man named Yossi Loya, who certainly was the leading coral reef scientist in Israel, and there's a huge pollution problem there. And that's why you've seen this die-off of the coral reefs and the corals in that region. At this point, it's extended all the way down to Raja Amput and the Egyptian coast. It's a big social problem, social economic problem of how you treat that. So we don't really know whether they have the capacity.
Last two questions over here.
We're making it worse.
Oh, sorry, Jeremy. Yep, there
Jim Van Etten from the University of Nebraska. I wanted to just put a plug in for viruses that was in the ocean. And it had always been assumed that there were about 100 virus particles per mil of ocean water until the early 1990s, when 3 groups independently showed they missed it by about 10 to the 7th logs. So they have done some fun calculations that there may be as many as 1 times 10 to the 31st virus particles in the ocean. And if you assume an average size and put them end to end, this spreads out to, I think, 10 to the 4th light years. So obviously one of the questions would always be, what kind of changes do we see with viruses, with all these changes that we're seeing in the ocean?
I'll maybe take a short response to that. Viruses are really important vectors for evolutionary change. And so as we've been talking about adaptation for species that are under stress, viruses might be a really important part of exchanging genetic novelty amongst organisms to help them adapt. In case anybody is worried like viruses are bad, they also can do really good and important environmental things.
Thank you all very much. It was great. Regarding Beth's very compelling presentation, it seems like being able to accelerate observations tremendously is pretty essential. And it made me think of the fact that astronomers and physicists have been extraordinarily effective at, for example, seeing distant things, finding ways to correct for turbulence and so on. It could be maybe a conversation to have here of whether there, amongst the astrophysicists here, whether there might be tools that could be developed that would be useful for that.
Thank you for that suggestion. And yes, I think there's so many disciplines that are needed to come to the conversation about accelerating technology for understanding the deep sea. So anybody that wants to talk about it, please come talk to me. I'm happy to connect you. Thank you.
Fantastic. Well, you know, ocean is the last frontier, and so still a lot to learn about the ocean, but there's some need for urgent steps and action. And I thank Marcia for highlighting the oceans in this symposium today. Thank the assembled panel for all their insights, and yourself for your question, and look forward to the Academy helping advance the science and advocating for the actions, the policy changes and actions that need to be taken to address challenges to the ocean health so we can achieve that goal of a healthy ocean for all. So thank you all for your attention today. Thank you. And now we're late for lunch.